webinar which is presented by the RAmediKa and Biocatalyzer. The webinar is on the breakthrough without sacrifice, exploring the future beyond animal testing. Here we are actually going to talk about the innovative practices which are actually like made to remove the outdated practices, that is animal testing. Okay, because we believe that innovation doesn't need a sacrifice. We are going to talk about many alternatives such as medical devices, organ on chips, pluripotent stem cells, and over in silico modeling. Okay, so I will like to invite our speaker from Biocatalyzer, Mr. Chandrajeet Dhara. I would like to introduce Mr. Dhara. So basically, Mr. Dhara is a final year BTech biotechnology scholar at the APJ University, Gurugram, Haryana, India. He is currently serving as a member of American Association for Cancer Research and American Society for Clinical Pharmacology and Therapeutics, and also he is an esteemed member of British Society for Antimicrobial Chemotherapy. Mr. Dhara has expertise in cancer genetics, nanotechnology, and medical immunology. He volunteered as a peer reviewer in the Curious Journal of Medical Sciences at the Springer Natures Group in oncology, internal medicine, and medical immunology. Mr. Dhara has published several research papers in international reputed journals, which is indexed in Scopus and Sciences, also in PubMed. One of the example is Spondius publication, Elsevier, and Bentham Sciences. We all know these journals; these are very reputed journals, and Mr. Dhara has like doing great work in the research field, and he is a co-founder in the Biocatalyzer. Now, I would like to hand over my mic to Mr. Dhara to please continue this session and please present your esteemed point of view upon the alternative of animal testing. Thank you.
Thank you so much, Mr. Harsh, for your great introduction. First of all, a warm welcome to you all for this amazing and insightful session on breakthrough without sacrifice, exploring the future beyond animal testing. So, before beginning my presentation, I would like to know, have you ever thought that why the world is now moving towards alternatives to animal testing?
I believe
World,
it's because we feel that animal testing is unethical.
yes,
It's they are
animal
also a
testing
living being, I
is,
guess.
yes, to conserve the nature. Ultimately, if all the animals or like the in vivo models or the animals will be tested uh, on the, for the drug development or biomarker development, then there will be one time where all the animals which are being tested till date will be an endangered species, right? Ultimately, the whole ecosystem will be hampered because ultimately the food chain will be hampered and later the ecosystem. So many ethical considerations arise during animal testing. That is why researchers are now moving towards finding the alternatives to animal testing, like uh, induced pluripotent stem cells, organoids, organ on a chips, lab on a chip, and furthermore. So I'll discuss in my presentation, I will talk about the role of induced pluripotent stem cells as an alternative to animal testing. So first of all, we will know about what are induced pluripotent stem cells. They are immature cells which are produced from skin or blood. Then they are reprogrammed into an embryonic like pluripotent state that creates an infinite supply of any human cell which are required as an therapeutic application. For example, induced pluripotent stem cells that can be induced to become beta islet Langerhans cells to cure the patients who are suffering from diabetes. Then blood cells to make fresh blood, fresh blood uh, for the cancer patients who are suffering from leukemia, or neurons to treat neurological problems like Parkinson's disease or Alzheimer's disease. So now the first step to make or design a immunopluripotent stem cell is first is the reprogramming. Reprogramming, scientists are actually reprogramming the cells that have undergone differentiation, like such as skin or blood cells that revert into an embryonic like state. The resulting cells are called, as we know, induced pluripotent stem cells. The patients, the biopsy samples are taken. Then they are cultured as an adult somatic cells. Then reprogramming. Then there are many reprogramming factors, such as some are integrating factors and some are non-integrating factors. Integrating factors, for example, retrovirus or lentivirus. Non-integrating factors such as episomal factors, mRNAs and microRNAs, proteins, exosomes. These are some of the reprogramming factors that are helpful for for programming that biopsy or adult somatic cells into the immunopluripotent stem cells or induced pluripotent stem cells, such as hematopoietic stem cells, neural cells, myocytes, adipocytes, beta islet Langerhans, hepatocytes. Then, though these pluripotent stem cells have an application in various avenues of biotechnology drug development, like cell therapy, disease modeling, drug testing, and many more. So now this is a uh, stained images of some of the induced pluripotent stem cells. First is the alkaline phosphatase, the A. Second is the OCT4. The OCT4 is a transcription factor, also known as, it belongs to the POU5F1 protein family that is encoded by POU5F1 gene. It is involved in self-renewal of undifferentiated embryonic stem cells. Third is the SOX2, SOX2. SOX2 is a transcriptional factor that plays a important role in the development of many tissues and organs during embryonic development. Third, fourth is the Nanog Alexa 488. Alexa is the dye. This Alexa 488 and CY3, these are the dyes which are used to stain these cells for the microscopy images. So this is the homobox transcription factor that plays a role in regulating the pluripotency of uh, embryonic stem cells. These are expressed in downregulated. Uh, these are expressed in pluripotent stem cells and downregulated during differentiation. TRA is a gene that in bacteria, in bacteria in and in Drosophila melanogaster, and it encodes for the T cell receptor alpha protein in the humans. Now we move to the following uh, applications of induced pluripotent stem cells. As first is its application lies in toxicology testing. Second is the in the drug discovery. Third is the drug disease modeling, and fourth is the precision medicine. Ultimately, the precision medicine, it has to work, because ultimately, currently, our world of medicine, medical science, is moving towards the precision medicine, designing precise therapy or personalized therapy for the patients according to their genetic profiles. So next we discuss about what is the uh, what you say, the assay of complete working of an induced pluripotent stem cell. The first process before after the designing or developing of immunopluripotent stem cell for physiology testing or something like drug testing. First of all is the assay development. In assay development, there are differentiations or multiple cloning is done, and they are done as three clones per genotype, which are established as a cellular endpoint, which represents a disease relevant phenotype. Phenotype should be robust, it should be reproducible, and it should be amendable. Since screening clones libraries are typically contains the compounds which are dissolved in DMSO, this phenotype, this phenotype should be DMSO insensitive. Then the library selection. The library selection, it should be the chemical screens have opted to use repurposing libraries which contains well sorted, well annotated targets, favorable drug like chemicals to enable rapid transmission into preclinical animal models and human patients. And then the screening of the dose. The screening of the dose, it is possible to perform the screen using multiple clones, and sometimes the typical screens use a single screen at a single dose. Dose can be between 1 micrometer to 10 micrometer. Then the secondary screening is done. In the secondary screening, the dose response, which is measured in EC50, and biological replicates are very important. Then the orthogonal and non-biological reagents are used for alternative reagents or reagent sources to vital to prevent the false positive results. Of course, when we are testing some drugs and all, so false positive results are very much we are prone to get. So the compounds that can be mislabeled or degenerated in library preparation that can mislead the cell or the screening list. Then for that, the orthogonal markers, in orthogonal markers, it could be antibodies, chemicals, or labels that are employed to reduce the compound induced to positive phenotype samples that are irrelevant for the disease, basically. So where there is a case study, there is a QT syndrome modeling here. The significant application of iPSCs have been seen. It's a cardiac disorder which can lead to arrhythmia, life-threatening arrhythmia. So researchers have developed cardiomyocytes, which are the heart cells, were used uh, for this study. These drugs, including potassium channel blockers and calcium channel blockers, were tested on these iPSCs, which actually the results, excuse me, yeah. So in this study, the potassium channel blockers and calcium channel blockers were uh, tested on the iPSCs derived cardiac tissues. These actually demonstrated that they reliably reproduced the normal cellular phenotypes, which are associated with long QT syndrome that provides insights into drug efficacy and safety without the need of animal models. So this approach not only validates the use of induced pluripotent stem cells for drug screening, but also highlighted their ability to mimic as human disease conditions. Now another case study which is described as chemotherapy-induced cardiotoxicity. It is for the patients with breast cancer and and which are the cardiotoxicity symptoms after the treatment of a chemotherapeutic drug, which is doxorubicin. These iPSCs were differentiated into cardiomyocytes to evaluate their responses against this drug. This study found that the patient-specific cardiomyocytes could be accurately predict which patients could suffer from cardiotoxicity due to the administration of the doxorubicin, which is on the chemotherapy-induced cardiotoxicity. It has been tested on the patients with breast cancer, and patients with breast cancer who have been administered with doxorubicin. Doxorubicin is a chemotherapeutic drug. So scientists have developed a immunopluripotent stem cell, uh, uh, which is cardiomyocyte derived induced pluripotent stem cell derived cardiac tissues, which are cardiomyocytes, to evaluate their responses to the drug doxorubicin. And this study found that this patient-specific cardiomyocytes could accurately predict which patients would suffer from cardiotoxicity due to the administration of doxorubicin. This capability allows for the personalized treatment plans, where the clinicians have the ability to adjust the doses based on the predicted drug response derived from the patient-specific induced pluripotent stem cells or immuno-induced pluripotent stem cell models. Next is the global market, about how big or how giant the global market of pluripotent stem cell production is. So the global induced pluripotent stem cell market would reach around $3.8 billion by 2024. And the market researchers also projected that it will reach a volume of $89.5 billion by 2031. And the next big prediction by the global market or market leaders, which are actually developing pluripotent stem cells uh, by about 2 decades, and research is going on, they have predicted that the market will establish from $1.48 billion in 2023 is anticipated to grow a capital annual growth rate to about 9.44% till 2030. So we can imagine how the application of induced pluripotent stem cells have been in the medical industry. We can imagine, right? It is a big market now. It is going to be a big market. So yes, it the market size and the applications and assays, everything can clearly draw attention of researchers, normal uh, human beings, everyone in this uh, planet, that the immunopluripotent stem cells or induced pluripotent stem cell and models are actually the future and could be a possible alternative to the animal testing. As it is easy, as it is convenient, the researchers are going on to make it a cost efficient treatment plan or assay plan that will open the door for precision medicine, as I have discussed. And precision medicine, precision healthcare, personalized treatment is the future in the medical science. So with this, I conclude my presentation, and I'm open to the questions you have. Thank you.
Thank you so much, Mr. Dhara, for your kind words. Literally, it was such an insightful that how pluripotent stem cells is going to rule the market in future. Seriously, we all are excited, and I believe that many healthcare professionals, future aspirants, are also with us, and they are also looking this pluripotent stem cells as a very innovative thing to work upon them, and we can definitely see the more alternatives upon the animal testing. And we are open for questions. If anyone has questions regarding this, please feel to ask anything. We are open to all type of questions. And even if your mic is not working, so you can put questions on the chat box. Okay. So Yeah, so first of all, we have a question from Ms. Archita Ranjan. She is asking that how can induced pluripotent stem cell based model be used to study complex disease like degenerative disorders or cancers? Such a nice question. Thank you so much, Archita.
It's really a very good question. So for the neurodegenerative diseases, uh, induced pluripotent stem cells derived neurons. There are neurons which are designed for the treatment of Alzheimer's disease, which are used to study the formation of amyloid plaques in the brain and the neurofibrillary tangles. So neurofibrillary tangles and formation of amyloid plaques are actually a key key factors of causing Alzheimer's disease. Second, the modeling of Parkinson's disease. Parkinson's disease, for the Parkinson's disease, induced pluripotent stem cell derived dopaminic neurons that are used to investigate the loss of uh, Mr. Harsh Shah, who is a final year MTech biotechnology scholar, and he is currently pursuing his master dissertation in medical device regulatory devices. Uh, Regulatory Sciences, I apologize, at the Sri Chitra Tirunal Institute of Medical Sciences and Technology, Thiruvananthapuram, Kerala. He is an active member of the Organization for Professionals in Regulatory Affairs, TOPRA, and his interests are in medical tech, regulatory affairs, and bioethics. He has been awarded with Student of the Year for National Services Scheme. He has been in the panelist at the Times of India. He is thrice inter-university debate champion, and he has published several research publications in the Journal of Oncology and CARV publications. Has served as the vice president and district administrator at Rotary International, and Mr. Shah has worked with some prestigious institutions in the country, such as IIT Delhi and DDU GKY, Government of India. So I welcome Mr. Harsh Shah, who is the founder of RAmediKa, would share, who will share his thoughts about the alternatives of animal testing models, like like I have shared about induced pluripotent stem cells. He will share about the organoid models, and beyond. The, the platform is all yours, Mr. Harsh. Thank you.
Thank you so much. I am audible, right? Okay,
Yes,
okay,
yes, you are.
okay. Thank you so much, Mr. Dhara, for such a nice introduction. I got flattered. So let's start with the presentation. So basically, ladies and gentlemen, I'm going to talk about the organoids and beyond, like organ and chips, okay, and beyond its future. Like we are actually talking about the future of animal testing. Okay, just a second. Yes, now it's visible. Yeah. So before I jump into the like depth of my presentation, we have to first understand that why it need to stop. Okay, because Mr. Dhara has talked about the like pluripotent stem cells, many alternatives he have discussed, but there is a question that why we need to stop, why animal testing need to be stopped? Because since history we are using animal testing. Now, why? What happened that we are going to stop it? So basically, there are four major points. First of all, ethical concern. That is very like obvious that ethical concern is one of the most prominent reason for stopping the animal testing. The second one is unreliable result. Yes, you will surprised to like know that animal testing has given unreliable results. So many unreliable results. We will discuss these things. Then we will look about the lack of validity. Well, yes, you will be very like curious to know how animal testing is actually lacking the research perspective in testing and all, and it is actually causing harm to human beings. Now, at the last but not the least, we will talk about the regulatory opportunities. What are the regulator point of view on the alternative of animal testing? So we will discuss each and one topic one by one. We'll first talk about the ethical concern. So, as I discussed, that it is unethical to sentence 100 million thinking and feeling in a laboratory cage and like intentionally cause them pain, loneliness, and fear. Yeah, we all have seen in like many movies that how animals are put on the cages and like scientists do their experiment, and like it is very like painful to see this. And because of that, this is it. And because of that, many like reputed organizations such as the Turkish Journal of Gastrology officially has banned the publication related to study involving the experiment on animal testing in their pages. This is one of the most significant steps this is taken by the international fraternity. Moreover, in August 2018, poll conducted in the Pew Research Center founded that the majority of the US adult opposes the use of animal in the scientific research. This was published in 2018. Now, there is one more thing that if it is morally wrong to cause animal to suffer pain, then why we are why we are experimenting upon them? You can see on this figure, which is actually like done in 1997 by Dr. Jay Vakanti, where his team is growing an ear, you know, a ear on the mouse, and this actually model is called as the Vakanti mouse. You can see like how painful and like painful is to see this type of experiment which doesn't have any purpose in the modern fraternity. Okay, many of these experiments cause pain to animal involved or reduce their quality of life in other ways. But I just think about a question that it is justifiable to conduct experiments such as producing an ear on the back of mouse in the name of minimizing suffering. I guess this is not just like justification that given by many people that we are actually minimizing their pain. We are killing them in a very like low pain methods so that they can bear the less pain during these experiment. I think that it's not justifiable because we innovator believe that innovation doesn't need a sacrifice. That's why ethical concern on this matter. Now, second, we will talk about the unreliable result. So we all know that when we talk about the drug discovery, okay. So there are phases in drug discovery, right? First of all, we need to identify the disease. Okay, the disease of interest we have to identify. Then we have to isolate the protein or any other molecule, protein. Okay, so we have to isolate them to analyze the structure, their functionality. So it will take around 2 to 5 years. It will take. Now, after we are confirmed with that protein or any molecule of interest, then we'll go for the preclinical testing. Okay, we will find the efficacy of those drugs or those protein upon the animals. It will take around 1 to 3 years. Now, once we are confirmed with that testing, preclinical testing, we will get the safety standards that it is safe to use upon the humans. Now, we will formulate them and we will find the new investigational drug according to US FDA, and then we will go for a human clinical trial. Okay, now when we go for human clinical trial, it will take around 2 to 10 years. Then, after getting a safe result, we will file the NDA, that is new drug application, and after reviewing by FDA, it will approve. You can see from the phase of discovering a protein until the FDA approval, it take around 15 years for making a new drug to come into market. But if we specifically talk about the preclinical testing, it takes about 3 years to 5 years. Okay, sometimes 8 years even, because we are going to test in different models upon the animal. And imagine within the period of 3 to 5 years, how many animals are being sacrificed because of that. Not imagine if I reduce this number, if I reduce this number to very less amount, then it will be very helpful for us to actually do the experiment within 1 year or 2 years. Even the National Institute of Health report has says that 95% of every drug that pass animal test fails in human. That's a very huge number. That 95% of the drugs fails in animal testing. Even the NIH director has even stated that the extrapolated results from the studies using tens of millions of animals failed to accurately predict human responses. That's actually surprising. 90% of the basic research failed lead to any human therapies within 20 years. Now we will see some graphs because I am not like talking in such a very random numbers. I am talking with facts. Now this study, which has been published in Nature, okay, Nature is very reputed journal, we all know. So this study has been published in Nature in 2018, where they are saying that the failure rate of new drug using animal testing. Okay, now you can see the Alzheimer disease. 99.6% like testing has been failed for an like Alzheimer disease. For cancer, it's 96%. For HIV and AIDS vaccine, it's 100%. For stroke disease, it's 100%, and even for sepsis, it's 100%. The questions come why we are actually like thinking to do animal testing. If we talk about the specifically a disease, if we talk about now, if I talk about the Alzheimer, okay, so we see in phases because in animal testing we go in three phases, that is first phase, second phase, and third phase. Now, if you see on the first page, like first phase, so 72% of the result has been failed, coming to 92% in second phase and 98% in third phase. Imagine the amount of animal we are using in this process and the result we are getting. It's definitely an unreliable result because 99.6% out of 413 trials testing upon total 244 the total number of testing, they have shown that out of those 244 tests has been failed. Okay, so this actually show a very huge impact upon us that animal testing are not reliable. We think that animal actually have some like genetic similarity with humans, so it can show some reliable result, but the facts are very clear that it is not showing. Now, when we talk about the another point, that is lack of validity. So, as we discussed in that previous data, that University of Oxford scientists found that animal research is likely to produce like less trustworthy result. That is very true. As we see in this previous data, that it is like providing very less worthy result. Even the studies said that the studies are actually biased towards the animals, and when it's come to human, it not actually replicate that safety standards. Okay, now we will study, like we will actually see those cases. Case study we will see that how the reliable studies from animal testing is not able to replicate on human. So in the first case study we will see in 2016, where a Portuguese company developed a drug intended to help with mood, anxiety, motor problem related to neurodegenerative diseases. Mr. Dhara has also talked about the neurodegenerative diseases. Okay. So here what they have done, they have tested their drugs. Okay. And what they found that six men aged 28 to 49 experienced adverse drug reaction and they had to be hospitalized. Even one participant has been brain dead. Just imagine you are testing a drug which is actually been proven safe in animals. Now one of the participant has been brain dead. Okay, even a report has been like notified that despite dose like 400 times stronger dose has been given to human volunteers. Even like we all know that at the point of interaction with humans, with patients, scientists used to give many doses to check the actually the replication of drug, the wavelength of drug, how it is replicating in the humans. So sometimes they even give higher doses and it can like result into the death of that participant. We will see the another case study which was done in 2010 that the in the like Northeastern University they have found many tragic result where the like after the first infusion of 500 doses smaller than the animals testing, six human volunteers faced life-threatening condition involving multiple organ failure and they were admitted into the ICU. Even 5 to 6 participants had to remain in hospitalized for 3 months. That is huge after the initial dose, while the other were in comatized. Now imagine if you were a participant. Okay, you are actually volunteering yourself for this noble cause of drug discovery, and you are getting this type of news that these type of testing are not reliable. It can cause into coma. It can cause you brain dead. Then will you able to give your consent? Yes or no? I don't believe that I will give my consent. Okay, now we will go for another sector that is regulatory opportunity. So when we talk about regulatory opportunity, so regulator has very broad perspective. like for the animal testing. If we see the 2017 conference where the US FDA commissioner has given the statement that drugs that test safe and effective in animals fails in human. We should find ways to modernize our traditional approach. It's a huge statement given by the US FDA Commission that he's also stating that the drugs are not safe and effective in humans. They are failing in humans. And but despite being like regulatory challenges, regulators are like relying on the animal testing because they believe that the like alternative model, for example, organoids, pluripotent stem cells, are under the research. Okay, they are in the research, and that's why they are currently rely on those animal testing. But they believe that like in future, in near future, within 5 or 10 years, we will be definitely testing our drugs, even for medical devices or biologics, upon the alternative methods. We will see those alternative methods. Okay, so we are actually like I am actually going to talk about the organoids, that is organ on chips. Okay, so that's a very fascinating topic. I'm telling you. Okay, these are one of the topics which not let me sleep. Even I guess Mr. Dhara will not able to sleep because of this such a nice topic. Okay, so basically, organ on chips or organoids are microfluidic culture device that replicate the complex structure and function of living human like human organs. They can mimic the human organ. Okay, as you can see, it's like lung on chip. So it is made up of flexible polymer, for example, polymethyl, methyl acrylate, and polycarbonate. Okay, it contain hollow microfluidic channel aligned with the living organ cells and human blood vessels. So basically, they are actually like those type of material which are actually non-reactive. They don't do any type of like adverse event, and these are actually providing a 3D cross section of human organ to mimic that actual thing. Now we will see how they are actually working. Okay, we will see the example of lung on chip micro device, and we will see how it is working. Right. So basically, the multifunctional device, micro device, can mimic the human alveolar capillary interface. We know that we have alveoli in our lungs, which helps in respiration. Okay, so this is the like micro device they have made, which can actually replicate the alveoli of lungs. Okay, so the micro channels that are created to induce air and nutrient in the epithelial alveolar cells. So you can see the channels. Okay, so these channels are actually like passed with the air, and they have epithelial cell above those plate, which can actually respire with the help of those air. And the vacuum channels are also produced. See, because we all know that when we inhale or exhale, so our lungs expand, and also it come to like deformation. So to mimic that thing, they have actually make a vacuum channel. So when vacuum come, it will get stretched. Okay, and when vacuum will release, it will go into this normal position. But this is how they are actually mimicking the lungs. Now imagine in a micro environment how cells are mimicking the lungs. Okay, now we will see like how they have made this. So they have created a microchannel of size 10 micrometer, which is fabricated with the porous membrane of polydimethyl siolane, that is PDMS. The interwing membrane was coated with the like fibronectin and collagen, and the human alveolar epithelial cells and human pulmonary microvascular endometrial cells were cultured upon this flexible membrane. Now you can see this is the porous membrane which is actually been used for cultivating the lung cells. This is actually very fascinating, right? We are actually culturing those lungs and upon the micro channels. We will see another type of organ on chips that are available on the market. We have also gut on chips and immune on chips. So you can see on the gut on chips that how the villi which are present in our intestine is actually mimicking the intestine of our human. Okay, and how the organ chips for human like reaction where our human like immune cells is actually being attacked with such pathogens, how they are actually mimicking those things. They are making those type of micro devices. Okay, even some very big giants company, for example, AstraZeneca, Roche, Johnson and Johnson, even Pfizer, They are also working on the organ on chips. Okay, and they have invested many like billions of dollar money with the help of NIH and FDA to make these things into the market. Now we will see the graph of how organ on chips has been has been expanding in the market, because Mr. Dhara has also talked about how pluripotent stem cells are growing in the market. Now we will see how rapidly the organ on chips is growing in the market. It is approximately will be reached around 77 million US dollar with the current CAGR of 25.6% till 2020 like 35. This is where I believe that this is very huge. Okay, because the way organ on chips is expanding in the market will definitely replace the animal testing. Now we will see some landmark judgment of Supreme Court of our India, what they have given, that India is also supporting. So first of all, we have some landmark judgments of Tilkat Bahadur versus State of Arunachal Pradesh in 1979, where the Supreme Court has passed a judgment to ban the animals in the cosmetics. Okay, now we all know that we have a Drugs and Cosmetic Act 1940, where the cosmetics are also involved in that drugs. Now, government and research has or like have realized that animal testing has not been reliable for the cosmetic. They have found their alternative. So, with that note, government has banned the animal testing for cosmetic. The same we can do with the drugs and biologics within and also with medical devices. It's one of the like supporting case. Even the law commission has given report that in their 261st report in August 2015, that they have observed that we need an alternative for animals. Okay, because it is actually giving a cruelty. It is actually a symbol of cruelty, right? And they're actually emphasizing that to promote public awareness about the like use of animal testing, they've actually recommended various authority, even the regulatory authority, that is CDSCO for the India, to actually look about the alternative of animal testing because of various ethical concern, right? Now, coming to the conclusion, okay. So basically, what we have learned that the statistics makes very clear that animals are not appropriate for human surrogate for biomedical research. Even European Union, UK, Belgium, Austria, Sweden, and many countries like New Zealand, Japan, India, they are banned the animal testing or they have limited the use of animal testing. Okay. And even like moreover NIH and US FDA, which are the prominent player in this field, they are also providing many funds. Even India also, we are providing many funds for non-animal testing like technologies. Even like I am associated with Suchitra, so Shichitra is also working upon the organ on chips, that is the alternative for animal testing. And multiple review has been documented that the animal testing has shown a great failure for human healthcare. With that note, I would like to say that we can't change history, but we can create a better future. Because humans are just one species among many. It is the species to believe that we have right to experiment on other animals using the misguided excuse that it might help human patient. Okay, animal aren't we definitely we all believe that animal aren't object. They are also an individual like us. When we look into their animal eyes, there is someone, not something. Okay. Looking back to us, someone who feels hunger, thirst, pain, fear, joy, and love. Because many of us has pets, we all know that what kind of bonding we have with animals. Now imagine if I take your pet to do experiment, you will not allow. Definitely will not allow. Okay. So with that note, I would like to rest my voice, and these are the references that I have used for my presentation. And thank you for attention. I believe that it has been a great session with Biocatalyzer and RAmediKa. And if you have any question, please feel free to ask. We are open for the questions.
What an impactful and data-oriented presentation, Mr. Harsh. I hope everyone can't just couldn't have just uh, diverted from their eyes from this presentation. Definitely, it was amazing. So I would welcome everyone to for the question session. So I would welcome first question. We have got a question from Keshri Sinha.
Yeah.
Uh, she has asked a question on, is there any law about stopping the animal testing,
Okay.
or have any petition filed in the court about this?
Okay, so first of all, thank you so much, Keshri, for asking such a very insightful question. Okay, so as I mentioned in my PPT, that the even the Law Commission of the India has put it their point of view in the view of animal testing. They are actually willing to have an alternative of animal testing, and even they have instructed the CDSCO to go for an alternative. I guess this is one of the biggest achievement that they have Indian judiciary have done for the alternative of animal testing. So I can see Archita has also asked a question, but Keshvi, coming to your question that any petition. So definitely we have many petition filed regarding the alternative of animal testing, and I was talking about the that how the Law Commission of India is actually putting their efforts to make an alternative. Even the CDSCO has been like facilitating many type of grants. Even the ICMR, that is the Indian Council of Medical Research, they're actually flourishing with lots of grants in crore of amount to actually provide an alternative for animal testing. So even like I am in a research institute that is a national important, that is Sri Chitra Technical Institute of Medical Science and Technology. Even Sri Chitra is also providing a huge amount of support for the animal testing. Okay, they are actually working upon the organ on chips that I have discussed in my presentation. So yes, I can definitely say that we are actually upon that path that within 5 years, I believe, hardly 5 years, we will definitely replace the animals as we are replacing the petrols. You know, we all know that how Nitin Gadkari, our minister, honorable minister, has been visioned to replace all the petrol vehicle with the eco-friendly fuels. With the same way, our science fraternity is also working upon that way to replace all the animals with the its alternative that is sustainable and that is ethical. Thank you. Now, Archita was also asking that question. Archita, can you please rewrite those questions? Ankit,
In the meantime, there is one more question from Ankit. What is the cost difference between organ chip technology and traditional animal testing method?
okay, so Ankit, thank you so much for asking such a like beautiful question upon the economics. Okay, so basically, if we see the costing, animal testing is actually very costly. Okay, because as I showed in the like presentation that how it is taking too much time for in especially in drug discovery, it take around 5 to 8 years. Now imagine you have to maintain those animal in a good condition. Okay, you have to give them regular nutrient, you have to maintain them for getting a reliable result. It will cost around billions of dollars. Okay, and in comparison to the organ on chips, or we can say pluripotent stem cells, even the in silico models, they're actually very less costing, because once you develop those medical devices, now we can actually put continuous research upon that. We can use for multiple research because it is actually mimicking the human organ, right? So with comparison with those animal model, I definitely believe, and it is actually fact, that how organoids had been used for alternative, and it's very cost effective, and that's why the market is booming. You know, I have shown in a graph that how the market is booming. It has reached around 900 million US dollar. Okay, that is huge. I guess I have answered your question.
I would request Archita to rewrite her question if she is in the meeting.
Yeah, she have actually righted the question that if animal models are not reliable, then why we are still using for testing purpose? Or definitely you have pinpointed such a nice question, Archita, that it's very obvious that we are talking about animal alternatives, right? We are talking about its various method. Okay, even Dhara has also talked about, we have also talked about, but why we are still using animal testing? The answer is very simple. First of all, it's regulatory foundation. Okay, because regulators have been not updated in their laws to use the alternative methods for their testing. Okay, just a second, guys. Yeah, sorry for the inconvenience. Yeah. So I was talking that first of all is regulatory foundation. Okay. Now the second thing is, see, when we talk about animals, so what animal is actually giving us? So animals are actually genetically identical to human. If you talk about mice, okay, like mice we are using, hamsters we are using. So those mice are actually 99% similar to human in genetic term, right? So with that note, we are working upon the organoids. So what we are working, we are actually taking the in silico model, that is computer-based model. Okay, because genetic code is nothing, it's just a code, biological code. Now, if we use the algorithm, okay, if we use the computer algorithm and we incorporate with the organoids, then it will definitely mimic the natural process what animals are doing, how they are naturally reacting upon those new drugs. This is the one of the like experiment that had done by US FDA with one of the renowned institute, that is Harvard University. They have published their research in the Frontiers, okay, where they have shown how the successful result by the in silico model and the organoids has been like shown 98% reliability success rate with that thing. So I guess you have got your answer, Archita.
In addition to this answer of my colleague Harsh, I would I would like to add some more points. I would like to add the point of integrating artificial intelligence like machine learning and uh, machine learning and deep learning models into the organ on a chip or induced pluripotent stem cells and these organoid models to increase its efficacy and to find where whether these models would work accurately and precisely as the mimicking model of that particular disease. So integrating artificial intelligence in current times, it's very important and it's still going on. And I am into that artificial intelligence working into deep learning and machine learning models for the early detection of cancer. So I am researching into this. So I think I am capable of answering your question and a slight addition to my colleague's answer.
Yeah,
So
definitely,
I hope
definitely.
it helps you.
Okay, there is one more question, I guess, is there. Can you see that question?
The
Yeah,
yes, yes.
Nihal
Nihal's
is
question, can we trust 100% on these organ models as it is not possible to replicate human body in full function?
okay. Okay, so Dr. Nihal, thank you so much for asking such a very insightful and curious question that can we trust upon 100%? See, like I will first of all say an ironic answer that we even can't trust human, 100%. You know, even many drugs and many tested, which are actually being tested upon the human trial, has been shown a very like what I can say adverse reaction upon the general population. Okay, so but with your answer, I can definitely say not 100%, but more than 97%. It has been shown an exact replication of the human models. Okay, because as I show, if I like, present my slide, just a second. Okay, if this is visible, right? It's visible. Yeah, okay, it's visible.
Yes, yes, it
So
is visible.
okay, so now you can see this. So I was talking about this image, lung on chip micro device. So basically, what we are seeing here, that with the help of epithelial cells that are present on our lungs alveoli, they are actually cultured upon those things. Okay, and with the help of micro chambers, air has been flowed in those chambers so that nutrient and air can be supplied regularly. And even we can see that with the help of vacuum, they have created a vacuum channels. So what a vacuum are doing? So we all know that when we create a vacuum, so vacuum actually create a pressure which make those chamber flexible and unflexible. Okay, they make us stretching and elasticity. So because of the elastic movement that actually happens in our lungs also with the help of diaphragm, the animal that these micro device model are actually replicating the exact copy of lungs. I hope you got your answer, Nihal.
And actually, Dr. Nihal sir's question is quite intuitive, like that the researchers are also working on the precision medicine, which are integrating into the organoids and beyond, and different organoid chips and induced pluripotent stem cells. Like I have mentioned in my presentation, also the integration of precision medicine, which is actually developing the therapy according to the genetic profile of the particular patient. So it is the patient's genetic profile which is helping to design or developing that therapy. So there is a complete chance of relying to the organoids or these next generation treatment methodologies or testing methodologies to 100%, as it is precise, it is accurate to that patient,
Yeah,
right?
definitely.
We can think about it.
Definitely, I would like to state, and I would like to add one point. Like you all know that there is an news has been recently come that human genome project has been done in India. Okay, where the 10,000 volunteer Indians have given their genome, full genome for research purpose. Now, what this is actually doing with the help of you like genome with the reference genome of our India, so we can actually make a precision medicine. Okay. So when we do animal testing and when we come into the human trial, so with the help of those data, because of the precision medicine, we can actually provide a precise targeting delivery system on the humans so that we can actually neglect the adverse event. Okay. So this is the one of the beneficial that how like genetic engineering and how biotech and how healthcare system are working with very significantly to solve many questions. Okay, thank you. Yeah, thank you so much, Nihal, for such asking such a nice question. And if
Thank you so much, Dr. Nialso.
yeah, thank you so much, Archita, Keshvi, Ankit, for asking very nice question and making this session very interactive.
Thank you so much, everyone, Archita, Keshvi, and Dr. Nialsa, Ankit, everyone for staying back for the Q&A session and encouraging us to answer with our experience or with our studies or research. So I would I would express my sincere gratitude to RAmediKa uh, for this wonderful collaboration with Biocatalyzer, and I would be thankful to you all that you have joined, you have spent your 1 hour or 1.5 hours of your Sunday to come and join this session and encouraging us uh, to express our knowledge or share our knowledge about this alternatives to animal testing uh, with you all. So I am looking forward to more such sessions with you all, and your participations will be highly appreciated. And I thank Mr. Harsh that he has supported me all through. Thank you so much.
Thank you so much, Mr. Dhara. And definitely, I would like to say that thank you so much, like participant, for trusting upon RAmediKa and Biocatalyzer. Okay, see, we are on the mission to actually provide awareness and educate people regarding such a novel research and innovative topics. Okay, so thank you so much again for like being the part of this interactive webinar, and thank you so much, Dhara, for being the speaker of this event and gracing us. Thank you so much. Now we would like to end the webinar,
The pleasure is all
and
mine. Thank you so much.
thank you. Now we would like to end this webinar officially, and the certificate of participation will be provided like to all of you on the group itself. Thank you for being with us. Thank you. Now
In the
I will,
Google Drive. In
yeah,
the
on the
Google
Google
Drive
Drive.
within 1 week.
Definitely.